Welcome back to the FARA interview series behind the scenes at IARC 2019, where Kyle and I are taking you behind... Wait, Kyle's not here. My partner in crime is downstairs bringing patient panel experiences with Friedreich ataxia and other ataxias to a room of 400 scientists. This is the connection of people and science and the disease that makes conferences like this so amazing. So I'm here alone today with Ian and Clementina, and we're going to talk with you about frataxin. So let's get into it. Would you please both introduce yourselves to our audience and tell them a little bit about where you work?
Clementina?
So I'm Clementina Mesaros. I work with Ian Blair for the last 15 years at University of Pennsylvania, and I initially got involved in the frataxin work, and then today we will stick to frataxin. So, Ian?
So I'm a pharmacologist. I was actually trained as a chemist a long time ago and became a pharmacologist. And in 2014, I was asked to become the person working on biomarkers within the Penn Chop Friedreich's Ataxia Center of Excellence. And so it was pretty obvious to me, if you wanted a biomarker of Friedreich's ataxia, it would be frataxin.
Seems sensible to me.
Right. So 5 years ago, we began to develop technology to be able to measure this protein, which is pretty difficult because it's not secreted. So you have to measure it in tissue and in blood.
Let's talk about frataxin.
Okay.
First off, what do you need? What do you need to measure frataxin?
A very expensive instrument. So they cost about $750,000, and we were very lucky in that we managed to secure funding from the NIH and from FARA to buy one of these beautiful creatures.
A mass spectrometer?
Yeah.
Okay.
And so this generation of mass spectrometers has really revolutionized our lives. I've been working in the field for 40 years, and it made a huge difference to my life because for the first time we were limited by imagination, not by our technology. So it's been an amazing experience, not just for frataxin, but for many other proteins.
Let's get right down to the very base of this.
Okay.
What is frataxin? I think we've heard it. We probably know it's a protein, but what is frataxin?
So basically the only known protein that is lower in FA, that FA patients are having a lower amount of this protein, and that's why it's very challenging to measure it and to measure it accurately, right? So because if there will be any clinical trials where people try to increase frataxin, you will need to measure maybe just like a 20% increase in this very low abundant protein. So you need to have very, very precise assays to do this.
Okay.
So actually it's a very tricky question. What is frataxin? So the full-length frataxin is a 210 amino acid protein, but it gets processed in many different ways. And so one of the things we discovered, it can not only be made in the normal way, but there's a way of making it through an alternative splicing. And we discovered a new form of frataxin that's in red cells.
And this was the subject of your talk yesterday to the scientists
downstairs. It was sort of the
downstairs.
subject of my talk. That was the inspiration for what we then proceeded to do, because it's in red cells, which don't have mitochondria, so it immediately made us think that there may be an extra mitochondrial function for frataxin. So that's working in the cytosol and in the nucleus.
And let me pause you there, because I want to keep our audience coming with us. Your presentation yesterday was about a type of frataxin that exists in red blood cells, which is unique, right? I guess, because usually frataxin exists in cells that have nucleuses, that have DNA, that have cellular functions, that have mitochondria producing energy, and a red blood cell doesn't have any of those.
Exactly, but it has to come from somewhere.
Sure.
So obviously it comes from a precursor cell that makes it, because red blood cells can't make proteins.
Can I ask you one more question? You used a term that might be interesting to folks out there, an amino acid, a 210 amino acid chain. Give us the 10-second version of
I've never had
how
to
about
do
this?
that before.
What is an amino acid?
So you've probably heard of things like alanine and phenylalanine and all these sort of things. So it's the building block of all proteins. And so that's what creates such a molecular diversity, because you can arrange them in many ways. Whereas,
Are they like, for lack of a better description, like letters in an alphabet that if you arrange them in a certain way, make one word?
Right.
And if you arrange them in a different way, make a different word?
It's even more
Exactly.
complicated than that, because
I would imagine.
they can have appendages off them as well, which creates an even greater molecular diversity. And so that's what actually become my passion, because the term for this is actually called a proteoform. So there are probably 30,000 proteins, but when they're decorated with all these different things, there can be literally billions of different forms, and I think that's what gives a molecular diversity.
So we know that frataxin is a protein.
Right.
We know that it's produced in cells, and now we know that it's also in red blood cells. What does frataxin do in the body?
Well, it's very controversial, in fact. So we know it's involved in the assembly of iron sulfur cluster proteins, but it's in every cell. And as you know, in Friedreich's, only certain cells are affected. So there's it's much more complicated than just downregulation of the protein. And
Sure.
So,
But let me touch on that for a second. So we've heard from some of your research and from some other researchers that FA produces different levels of frataxin in different tissues. So there's a different effect in the heart than there is in the brain and spinal cord, than there is in the muscle and peripheral nervous system. This is what we're talking about now.
well, it's not hugely different. So there's something else that we don't understand. So that was actually the main purpose of that talk yesterday. You know, if there's a form of frataxin that's not in mitochondria, what is it, and how much is there?
And what does it do? And this is the work that you presented yesterday, and that your lab kind of continues to plow forward on.
So now, using animal models, we've shown there's a form of frataxin that's not in the mitochondria. So now we can circle back, and maybe that explains human physiology to some extent, because now we're involving much more than the mitochondria. And so I think that's what our research has opened up now. So it's going to be a very exciting couple of years. And especially with all the therapeutic approaches are really designed at upregulating frataxin in mitochondria. So now we have to be aware of the possibility of other forms that might have to be regulated. You agree?
Definitely.
Okay.
We've talked with other researchers about how their research will hopefully lead to clinical trials, and about how we want to design endpoints for those clinical trials, a way to determine what the medication or the therapy does in the body. My question, and it might seem simple, help me out, is this. If the end goal of all of this is to just increase the amount of frataxin that is produced in patients who have Friedreich ataxia, why don't we just directly measure frataxin as opposed to all these other difficult clinical endpoints? Why isn't that just what we do?
Do you want to have a go at that?
Because it's really difficult to measure. First, because they are very low levels in circulation, and second, because of all these proteoforms that Ian mentioned. So right now, I don't think that we are sure if we measure one proteoform that we have the right proteoform that we need. The body needs.
Of course, the other issue is you can't just take bits of people's brain and measure it.
Sure.
Or you can't take bits of their heart and measure it. So you've got to find a surrogate. And so that's why there's so much interest in trying to develop biomarkers. So if you can figure out what it is that frataxin, what's the next step, then you have a way of monitoring it. And we've done several things in that direction. We've been using platelets as a way of monitoring metabolism in Friedreich's patients. And we showed that OMAV, that Reata drug, actually normalizes metabolism in human platelets, and it helped to get the right dose. So that's an example of how we weren't really measuring the frataxin, we were measuring the downstream effects, which is a dysregulated metabolism, that then this drug normalized, and now we see that some of the clinical symptoms are also alleviated. So
you're measuring a thing you can measure, which you know is related to frataxin, which you can't measure. That makes a lot of sense.
Right. So the next, so the brain is much more challenging. So there was a paper from the Voyager group showing that they can measure very, very tiny amounts of frataxin in cerebrospinal fluid. So that might offer a way. But there may be other ways that we could get at that. Maybe some of the downstream proteins that are upregulated by frataxin are in CSF, and we can measure them. And one of the more crazy ideas I've had is using nasal epithelial cells, so taking them from the nose and looking at it there, because there actually is evidence that olfactory function is reduced in Friedreich's patients.
Olfactory is the ability to smell.
So that would suggest that frataxin is involved in smelling in some way. And so if you're correcting a deficiency in the brain, presumably you're also correcting something in the nasal olfactory cells. But it's been a hard sell to get people to want to do it. I had it done to myself, and it's not terrible.
it's a scientist who experiments. Right. Physician, heal thyself.
Right.
Let me ask you this before we leave.
Yeah.
Let's talk about naked mole rats.
Oh, naked mole rats.
All right. Help me out. I was told...
Should I tell you the whole story?
Not the whole story.
Okay,
we're on the clock, but
okay.
I was told to ask you about naked mole rats. So can you tell us about the naked mole rat and why it's important?
I took a friend of mine to a sabbatical in San Francisco where they're working on naked mole rats. And we had this extended journey back through Canada to Philadelphia. It took us 10 days. We stopped in Portland, and we were in a bar, and we were listening to the Beatles. And for some reason, I decided to look up the sequence of the naked mole rat frataxin.
At a bar in Portland.
Right. I mean, as you say, we're sort of not quite normal as people, and there's a program called Uniprot which has all proteins in it, and you can look at the sequence, the amino acids in any protein you care to look at. So for some reason that I still don't know why to this day, I looked up the naked mole rat frataxin, and the sequence is almost identical with the sequence of the human red blood cell frataxin.
The E-frataxin or erythrocyte frataxin.
Yeah, the erythrocyte frataxin. So at that point, our work just seemed like, oh, well, who cares? Red cells are fine in Friedreich's. This is some weird finding that doesn't mean anything. But then that made me realize this is important.
Yeah.
And so these are the most long-lived rodents on the planet. They live for 30 years. And, frataxin deficiency limits people's lives. So you put two and two together, and maybe there's something about frataxin that's not just neurodegeneration and cardiovascular disease. Maybe it's involved in something else, and it's that something else that we're kind of obsessed with at the moment.
Sure. Well, we're thrilled that you're obsessed with frataxin.
Well, one more thing.
Oh, okay.
What the creatures that live even longer are bats.
Okay.
So their sequence is even closer to human erythrocyte frataxin than naked mole rats.
Like vampire bats or?
Brandt's bats, the main ones. So there's a bat research centre in Ireland where they go to French chateaus and get the bats to come into their nets. And so I'm trying to get bat blood from them to see if it's the same stuff. So this is going to be quite a fascinating story in years to come.
Yeah, it already has been.
Yeah.
Thank you so much for all the work you do on behalf of our entire community. Thank you for sharing your time with us this morning. It's been a real pleasure.
It's been a pleasure for us. Thank you.
All right. Be sure to tune into our next episode of the FARA interview series, Behind the Scenes at IARC 2019. Stay tuned to additional episodes that are available on the FARA YouTube channel, on FARA Facebook page, on FARA Twitter, and FARA's LinkedIn resources. Until next time, thank you very much.